the other side of the world for a little while. And I trying to get rid of this screen that says recording in progress, and it's not allowing me to do that. Okay, got it. All right, sorry about that. So yeah, I grew up in a family of scientists and artists. So my father is a biomedical engineer and my mother is an artist. And this sort of left brain, right brain combination, I think really gave me an appreciation for the art in science and the beauty of biology and mathematics. And initially, I was very focused on mathematics as a young child and as a student. And I graduated high school. So I skipped a couple grades, graduated high school when I was 16 and started university, and was really focused on mathematics and biomedical engineering. And I thought for sure I would become a professor and that I would be doing mathematical modeling of biological systems for my career. I had no idea I was going to end up in toxicology. If I had known that, I would have paid a lot more attention in my chemistry classes. So moral of the story there is that you never know what you're going to end up doing. So pay attention in all of your classes. And I ended up working at a lab at Duke University in the biomedical engineering department as a research assistant. And I was accepted into a PhD program at Duke, and I was all set to go there directly from my undergrad when I was like 20 years old. But I had kind of a personal thing happen during the finals week of my senior year. My boyfriend broke up with me, and my best friend came to my apartment and she said, I want to go to Australia and New Zealand and I want you to come with me. Let's take a year off. And so I decided to do this crazy thing and take a year off in between undergrad and grad school. And my advisor at Duke was very kind and said, no problem. The money will be there when you get back. It's a 5-year grant. So go have fun and I'll see you in a year. And so we went to Australia and New Zealand. And in Australia, we just lived and worked on the Great Barrier Reef in a hotel, doing nothing even remotely intellectually interesting, but in a beautiful place. But so by the time we got to New Zealand, I was really starved for science. And so I went to the Center for Bioengineering at the University of Canterbury on the South Island of New Zealand, and asked if they would hire me as a research assistant. Which was pretty audacious. I had no contacts there, but I just marched into the center director's office with my CV and handed it to him and said, hey, I have a working holiday visa. I can work for you for 4 months. Will you hire me as a research assistant? And so I guess the moral of the story there is never be afraid to ask for anything, even if it seems totally crazy and out of left field, because you never know what opportunities you can be offered if you don't ask for them. And so they hired me as a research assistant, and I loved it there. And I worked on some really interesting projects on imaging of blood flow in the brain and understanding computational fluid dynamics approaches to being able to target, surgical interventions for people that had different vascular anatomy in their brain. And by the end of the few months that I was there, they offered me a targeted international doctoral scholarship to stay and do my PhD at this university in New Zealand. And that was a really tough choice for me, because here's this gorgeous country that looks like Lord of the Rings everywhere you go. It's just astonishingly beautiful and really nice people and very good research being done at this sort of newly founded Center for Bioengineering, but it's the other side of the world, and nobody's ever heard of it. And on the other hand, I have Duke University, which really, not quite Yale, but almost an Ivy school, right? And I've already been accepted into a PhD program there. So what do I do? Do I go with this sort of lesser known but very exciting opportunity? Or do I go with kind of the safe route and go back home to North Carolina? And I decided to kind of, take the road less traveled. I decided that we increasingly live in a society that is very performance-based. And so I thought, well, if I do great work and I publish in great journals, then it doesn't matter where I get my PhD from. Does it need to be Duke, or can it be this other university on the other side of the world? So I moved to New Zealand, and I lived there for 4 years. And it was one of the best decisions I've ever made. It was an incredible place, and I did really fantastic research, and I published in the American Journal of Physiology, really top-notch journals. Went to some amazing conferences, met a great network of systems biology modelers, and then after all of that, I ended up coming back to North Carolina to do my postdoctoral research with the U.S. Environmental Protection Agency Office of Research and Development. And I started at the same time as the EPA's ToxCast program, which is part of the federal Tox 21 research program that was just getting off the ground. And so it was an amazing opportunity for me as a new postdoc to basically walk into a sea of data. And they said, here's all of this data, please do something with it. And so I could apply my biomedical engineering expertise and my mathematical modeling background to start to understand some of this high throughput screening data that was being generated on, at the time, hundreds, and now more recently thousands of chemicals, and try and build these multi-scale systems models to help understand how those data could inform on environmental health risks being posed by those chemicals. So I guess, the moral of the story there is, don't be afraid to take that path less traveled, because I don't know, if I hadn't done that, I don't know if I would have been able to land this amazing postdoctoral research opportunity at the EPA, kind of coming from left field and having this really diverse background and training. So I did my postdoc at EPA, and then I went into industry for a while, not industry like you would think of in terms of chemical industry, but in like a contract research organization, still supporting the federal government as a contractor, and doing data analysis and leading computational toxicology work for the center that I now direct. And so I, started off as a group lead in that center, and then I got hired back onto the federal side to be the deputy director. And then for the last several years, I've been the director of the center. So that was a very long intro. You've been staring at a title slide for a very long time, but I wanted to give you some context about myself on a personal level and kind of how I got to where I am professionally. Okay, so jumping into the content of the presentation, I want to start with just a brief intro and overview of who we are. What is NICEDM? What is ICVAM? ICVAM was mandated by the U.S. Congress a little over 20 years ago to kind of establish this committee to coordinate among different federal agencies that either require or consider chemical safety testing data, and are very interested in moving away from a reliance on, slow, outdated animal tests that are potentially not very relevant to humans, because at the end of the day, we're not 140-pound rats. Our biology is different, often in the very, signaling pathways that make up the basis of disease mechanisms and etiology. So having this coordinating committee among these different federal agencies was seen as a high priority for Congress to make sure that we are optimizing resources and communicating among both regulatory and research agencies within the government. But it's a voluntary committee. So everybody who serves on ICVAM from all these agencies that you see listed on the screen here is contributing their time in kind. And we have some very senior people. So the senior science advisor for the US EPA is on ICVAM. The senior scientist in the Office of the Commissioner for the FDA is on ICVAM. So we've got lots of senior people helping to tell us what those agency priorities are. And then NICEDM, my center, is kind of the workhorse of ICVAM. So we have a budget of about $5 million a year. I have a group of about 22 people. And we work on projects that all these different federal agencies identify as priorities. So they'll come to us and they'll say, hey, we're really worried about developmental neurotoxicity. Like there's a huge increase in kids being diagnosed with autism and ADHD. And there's obviously this environmental component. And we don't have good animal models to try and understand what chemicals might be interfering with very sensitive neurodevelopmental pathways. Can you help us build new testing approaches and validate those approaches so that we can start making regulatory decisions based on, human neural cell assays or small model organism behavioral assays, et cetera. So all of this is outlined in this strategic roadmap that was actually published 5 years ago now. So we're celebrating the 5-year anniversary of this U.S. strategy and roadmap. And this kind of gives you a high-level overview of how ICFAM operates, how NICEDM supports ICFAM, and what we do. And it's, I think, very what's really important about this cover, and the reason that I put it up there, is kind of this iterative process that's represented by the figure on the front, which is the necessity for sort of constant communication among end users, so regulatory decision makers, groups such as myself, my own group that does validation of new approaches, and groups like many of you will work with or will go on to form yourselves, which are like academic groups and groups in like the biotech sector that are developing new approaches. So developing really cool new technologies that can be used to screen chemicals or understand potential chemical health effects, but making sure that those new methods are being developed with regulatory needs in mind. And so that's kind of the fundamental basis of how we operate, is having that sort of circular communication among the end users, the method developers, and the validation bodies. We have tons of different projects, a very broad portfolio of work. I won't be able to talk to you about most of them today, but if you have a particular interest in any of these individual subjects, please follow up with me afterwards. I also, get out your phone and scan the QR code and make sure that you're subscribed to our NICEDM News email list. And we have newsletters that come out about every week, maybe every other week, and they have lots of awesome information in there, like grant funding opportunities, jobs, PhD student opportunities or postdoc positions, or conferences like travel awards, new publications, et cetera. So really nice wealth of information there. And then also the ICFAM Biennial Report is a tremendous resource. It provides you just like a paragraph overview of each of the activities among all of the different federal agencies in the space of NAMs, or new approach methodologies. And then this image in the middle here is what I like to call the computational toxicology continuum. So when I came on board with NICEDM about 10 years ago now to lead their CompTox group, and then as I've ascended in the leadership of this organization, we need to increase our focus on computational resources kind of underpinning all of what we do, because we live in a world that is increasingly reliant on computers, and we rely heavily on technology to help us understand and predict human health risks from tens of thousands of chemicals. So this is a figure from a publication that we co-authored with some of our academic collaborators and the NIH Office of Data Science. And it really sort of represents that perspective on the essentiality of computational resources at every piece of the pipeline, from generating data to processing, curating, standardizing, integrating data, with the goal of enabling better models and tools to predict chemical effects and biological effects. And, I think it really demonstrates kind of how important those computational resources are to kind of support high quality data generation, sharing, and application to sort of connect those two sides of the spectrum and enable, trustworthy outcomes and decisions. And so I think a lot of people use this term AI, I don't want to say incorrectly, but they use it in a limited way, right? So my title here for this talk was using augmented intelligence to enhance environmental health science research. Most people think AI and they think of artificial intelligence. So this like process of building, a human-like intelligence, like an autonomous technological system, like a robot that might someday replace us. I think my definition is more comprehensive. And I should, caveat this up front that I didn't come up with this term, augmented intelligence. It was actually first published in the late 50s and early 60s in this series of seminal papers that I have, cited on the screen here. But I'm truly trying to socialize this term and get people to expand their thought processes around AI to really be better defined as augmented intelligence. So a field that leverages, artificial intelligence and machine learning, big data, natural language processing, mathematical modeling, data analytics, all of the computational tools in our toolbox to enhance and support human intellect. And really goes like beyond that typical perception of just let the machine do it, but actually use the entirety of our technological toolbox to improve and build upon our own expert knowledge and our intellectual capacity. And so I'll give you several examples throughout the talk of how we're trying to apply augmented intelligence. So one example here is developing what are called QSAR models, so quantitative structure activity relationship models. I think most of you are familiar, but sort of the basic principle is that if you have enough data on a diverse enough chemical set, you can apply machine learning to train models that can identify complex relationships between chemical features, so things like structural properties or physicochemical properties, and whatever endpoint you're trying to model. And those endpoints can be toxicity endpoints like endocrine disruption, or endpoints like environmental fate or distribution within the body, et cetera. And so using this augmented intelligence type of an approach, we first use an expert knowledge-driven method to curate training data. So we go out and identify large data sets, and we use our subject matter experts to look at the quality of that data, curate it to a high quality training set that we then share with the best computational chemistry groups around the world. So really trying to take a crowdsourcing approach towards building these models. We have excellent computational chemists in my group, but we recognize that we're not the only game in town. And there's plenty of other groups around the world that have their preferred set of descriptors or algorithms that they like to use. And so building ensemble models, so sharing the training data with all these groups and then having them give us back their models that they've built on that data, including predictions on external validation or test sets that we can use to evaluate the performance of the model. We then can combine the best models using an ensemble approach that leverages the strengths and compensates for the weaknesses of any single method. And then we get these incredibly robust models that can be used to predict toxicity across the entire chemical universe without having to do any testing. And when compared to the animal tests, are as good or better. And so we have an open access model suite of QSAR models called OPERA. It's freely available to the scientific community through the NIEHS GitHub and through a number of other avenues that I'll show you in just a second. I think some of the most exciting projects that we've done under the OPERA suite have been these international collaborative projects that are targeted towards regulatory endpoints. So we've done a couple around endocrine disruption, and then we've done one around acute systemic toxicity. I'll focus first on the endocrine disruption work. So this is a really exciting development. So in December, the US EPA published a draft white paper announcing the availability of new approach methodologies within the regulatory endocrine disruptor screening program. So they have this mandate to try and understand what types of chemicals in the environment might interfere with, the exquisitely regulated, very tightly regulated endocrine system in your body. So the balance of hormones that contributes not only to things like reproduction and development, but also to metabolic syndrome, many different types of cancers, and just your overall sort of homeostasis and well-being. And this is obviously of concern because the system is so tightly regulated in your body that, you know, fluctuations, minor fluctuations in the concentrations of different hormones can set off signaling cascades that have major biological effects. And a lot of chemicals in the environment look a lot like our endogenous hormones. So I'm sure many of you have heard of BPA, bisphenol A. This is an estrogenic substance. It looks a lot like estrogen if you look at the chemical structure, and it binds to your estrogen receptors, and it can cause, you know, lots of downstream signaling that's potentially adverse in your body. And so there's a real need to be able to screen chemicals in the environment for their potential endocrine disruption. And in particular, the models that I just talked to you about, so these QSAR models for predicting ER, estrogen receptor, and androgen receptor activity, are now acceptable by the US EPA in a regulatory priority setting and weight of evidence analysis framework. And that those are on the screen. Those are quotes from that white paper. So very exciting to see all of this hard work really actually being translated into regulatory decision-making and implementation. Another sort of effort that's been really, really important for us as we're trying to put these new approaches, these computational models into, you know, basically assess their readiness for regulatory application, is understanding this as good or better kind of benchmark for replacing the animal test. So you want to make sure that, you know, your new approach that you're proposing for regulatory use is going to be as good or better as the animal test that you're replacing. Well, to be able to do that, you have to understand, like, how good is the animal test? And in one of the other sort of crowdsourcing approaches that we did towards predicting toxicity, we were looking at acute systemic toxicity. So this is a test called the LD50. So the lethal dose at which 50% of the population would die. To put it in layman's terms, you take a group of rats, you stuff them full of chemical, and you wait for half of them to die. And the dose at which half of the rats die is called the LD50. It seems kind of barbaric, but it's actually an incredibly important toxicological value for so many different risk assessment applications. So the LD50 determines, you know, what kind of personal protective equipment you have to wear when you're handling a chemical. It determines where that chemical can be used. Can you use that chemical in industrial manufacturing, or can you use it in playground equipment or in, you know, household cleaners that people are going to be exposed to all the time? You know, it forms the basis for things like ecological risk assessments. So it's an incredibly important value in the toxicology community. And so there are like tens of thousands of chemicals that have had rodent LD50 tests run on them. And because of that, we were able to identify a couple thousand chemicals that had actually independently replicated LD50 tests. So different labs at different times had run that animal study, that LD50, on the same chemical. And so we could ask, well, how reproducible is that test? Because that provides a basis for comparison for any new approach that's trying to replace that test. Because if you think about it, it's not reasonable to expect that a new approach should do better at predicting this animal test than the animal test does at predicting itself. So the reproducibility of that animal test is a good quantitative kind of benchmark for the performance of these new tests. So we used a statistical bootstrapping approach to derive a data-driven confidence interval around those acute oral LD50 values. And then that provides a benchmark to be able to say, okay, our QSAR models trained on over 10,000 chemicals actually do very well in predicting those LD50 values, and they are equivalent in terms of the reproducibility of those in vivo animal tests at predicting themselves. And so we're applying this, we're working with the US EPA to apply this in a eco risk assessment perspective. So as I mentioned, LD50 is used all over the place. And one of the major uses for the EPA is to use it as a surrogate to assess risk to all mammalian wildlife. So before we started this project, I had no idea how complicated ecological risk assessments are. They look at so many different conditions. They look at like little tiny mammals eating long grass or eating short grass, and farm mammals eating long grass, and birds that nest beside streams that have effluent in them. I mean, there's all these different conditions that they look at, and they use the LD50 as kind of their benchmark value to understand toxicity risk across all these different components of the ecosystem. And so we went back and we looked and said, all these risk assessments that have been done over the last several decades by the EPA, what would happen if you substituted in the predicted value for the LD50 instead of the measured value? And long story short, it did really well. This is work that's in preparation for publication right now. But spoiler alert, we are pretty confident that the EPA is going to issue waiver guidance for, any LD50 prediction of like that's non-toxic basically. And that's good because pretty much all chemistries that are coming out, all new chemistries that are being registered with the EPA are engineered to be non-toxic to mammals. And so if you can just use the computational model and predict the LD50, you don't have to run the animal test as long as it says that it's non-toxic. And so that's where we're hoping that we'll get to within about a year, that the EPA will write that waiver guidance and we can stop running the LD50 test. We're also applying this to mixtures. So what I just talked about is really only applicable to like a defined chemical structure. So you have a single chemical structure, you run it through your computer model and you get a prediction. But what happens if you have a mixture, which is the vast majority of things that we're exposed to, like agrochemical formulation products, for example? Well, there's this approach called the GHS mixtures equation, or the GHS additivity approach, where you take a weighted sum of the components and use that to predict the toxicity of the mixture. And we showed that you could do that very effectively with the existing in vivo data. But then our colleagues at the US Air Force took it a step further and they said, well, what if we use CATMOS? What if we use that QSAR model, predicted the LD50 of the individual components, and then use the additivity equation to predict the LD50 of the mixture? And they basically showed that it was very effective to do that same thing. So again, you wouldn't even need to run the rodent LD50 test on the mixture. You could just use an in silico approach. So as I mentioned, these toxicity models are just a handful of models in a much broader suite of models that are contained within OPERA. So Kamel Mansouri, who's on screen here, is our senior computational chemist, and he maintains this suite and develops it out. And so you can see that we've got prediction models for all kinds of phys-chem properties, environmental fate parameters, things like plasma protein binding, so things that determine how chemicals distribute in your body, and different toxicity endpoints. And then we have a number of models under development. One area where we've put a lot of effort is into optimizing models for PFAS compounds. I know you all are very familiar with PFAS as a major emerging concern in human and environmental health. So these forever chemicals don't go away. They're absorbed by your body. Over 97% of the American population has detectable levels of PFAS in their blood, and they've been linked to a number of human health concerns. And so we are trying to optimize our prediction models to be able to predict these chemistries, which are traditionally very challenging chemistries to predict. And this was an independent assessment by a Canadian group that looked at a number of different computational models to predict properties for PFAS models. And in every endpoint, they found that OPERA was the best performing model for predicting PFAS. So you can find OPERA model predictions on the EPA CompTox Chemicals dashboard. You can find it on FDA's Precision platform. Most recently, you can find it as a plugin in the Organization for Economic Cooperation and Development, the OECD's QSAR toolbox, which is used by regulatory authorities worldwide. And then my personal favorite, nearest and dearest to my heart, is you can find these predictions on our integrated chemical environment. And so I'm going to spend the next few slides talking to you about what the integrated chemical environment is. And if you were one of the students that was in my lecture that I gave last month, some of this will be familiar to you, but hopefully will be a nice little refresher. We did just release ICE version 4.0. So we released that just ahead of the Society of Toxicology annual meeting. And ICE is really intended to democratize access across the entire scientific community to high curated quality data and computational toxicology workflows. So we want CompTox to be the domain of all environmental health scientists, not just folks who know how to write code or to program, but we want it to be really applied across researchers and regulators alike. And so you can find in ICE, you can find lots of data. You can find things like reference chemical lists that have been used to validate new assays. You can find curated information on the high throughput screening data from the Tox 21 and ToxPass programs, where we've annotated those assays with defined terminologies for mechanistic targets and toxicological modes of action. And then you can find these computational workflows that do things like allow you to run virtual animal studies or virtual human studies and predict internal concentrations, or look at a group of chemicals and say, you know, what is the distribution of, like, where do these chemicals fall in the broader chemical universe, or compare two lists of chemicals and say, how similar are they? Or where do they appear in consumer products, et cetera. So just to give you sort of a little bit more detailed view, here is a list of some of the data that we have in ICE. So we have a number of different experimentally derived parameters for physicochemical properties or ADME properties. We have data on acute toxicity, cancer, developmental and repro tox, different topical toxicities, endocrine related toxicities. As I mentioned, we have a curated version of the high throughput screening data from the Federal Tox 21 Research Program. And then we have lots of in silico model predictions from OPERA. This actually should be 1 million plus structures, because in the new release, we included a new version of the OPERA predictions on over 1 million defined structures. This gives you just a beautiful kind of snapshot of the different tools within ICE. So you can explore these Tox 21 data sets whose assay targets are linked to organ systems in the body and mechanisms of toxicity. And as a user, you can actually interact with the actual concentration response data from individual assays. Or you can overlay concentration response plots from, multiple different assays on one chemical, or multiple different chemicals on one assay. You can do that in 2D, you can do that in 3D. You can filter the data based on a variety of parameters like mechanistic targets, or what cell types it was measured in, or what bioactivity level. You can look for similar chemical structures. So maybe you have a chemical that you're interested in that doesn't have very much data, but there's other structurally related chemicals that do have data. So you can, again, combine and rearrange filters to refine those results and, look for more data-rich compounds that can help inform on your chemical of interest. As I mentioned, you can run virtual animal studies or human studies. So you choose parameters like the species, the dosing route, whether you want inhalation dosing or injection or oral dosing. You simulate, different time frames, repeated dosing, and then you can get outputs that give you predicted concentrations in different tissues in the body, so at steady state, or time course of the concentrations of different chemicals in different tissues in the body. You can also run that in reverse. So you can say, oh, I have activity concentrations in these assays that I know are relevant to certain biology that I'm interested in. What would I have to be exposed to for those chemicals to reach internal concentrations where I'm seeing activity in my assays? And that's called in vitro to in vivo extrapolation. And I'll give you a couple more examples of that in a minute. And then the last workflow that I've highlighted here is where you can look. So we have a full instance of the US EPA's consumer product database on our back end. And so you can look where your chemicals of concern appear in consumer products, and then you can compare those with patterns of bioactivity across different groups of mechanistic targets to sort of further link exposure to potential toxicity. A number of new features in version 4.0 that we just released a couple of weeks ago. We have a beta version of a new, like, complete redesign of our search results interface with interactive visualizations. We also have additional enhancements to those PBPK and IVIVE models. So we now have a gestational model. So you can look, if you're really interested specifically in developmental toxicity, then you have a new gestational model that looks not only at the concentration in the mom, but in the predicted concentration in the fetus as well. And then we have additional exposure predictions, which I'm super excited about. So there's a completely new data set in ICE where we've incorporated not only measured exposure data from NIEHS biomonitoring studies, but also predicted exposures from the US EPA's ExpoCast project. And so you can actually do that overlay. So you can start with in vitro activity concentrations, do the IVIVE, say how much would I need to be exposed to achieve internal concentrations where I'm seeing activity in my in vitro assays, and those equivalent, administered doses, you can actually then overlay that actual human exposures or predicted human exposures and say, well, how far off am I? Or do we have really small margins of exposure here? And therefore I'm more concerned about this chemical, et cetera. Okay, I'm going to kind of start to go a little faster through some of the rest of these slides because I know I'm pushing up on time here. But a specific example of how we've used ICE and this technological toolbox to augment our intelligence is in the area of predicting environmental chemical cardiovascular toxicity. So we know that cardiovascular disease is a very prevalent human health issue in the US and also globally. Chemicals aren't really screened for cardiovascular effects in your typical toxicology studies. And so we've leveraged the Tox21 high throughput screening data and used expert biological knowledge to map those different human cell-based assays to what are known as cardiovascular failure modes that have been defined by the FDA. And then we create these cardiotox pies where each slice of the pie, so that's that pie chart that you see right there. So that's the full pie chart. And then each of these cardiotox pies represents the activity of a specific chemical and helps us visualize which of those chemicals have potent or specific activity against critical cardiovascular related targets. And then we can use cheminformatics approaches to identify clusters of chemicals that are enriched for that cardiovascular activity, and then apply that IVIVE approach to estimate external exposures that would be needed to achieve internal concentrations in the body where we're seeing that cardiovascular relevant toxicity. Then compare those estimates to chemical exposures from biomonitoring studies. And that allows us to identify priority chemicals that have small margins of exposure, or in some cases where the predicted toxicity and the actual human exposure levels are overlapping. And so some of those were very confirmatory. So air pollution components, for example, we know that air pollution is inextricably linked to cardiovascular disease, and doing this, this whole workflow, immediately some of those air pollution components rose to the top and said, yeah, absolutely. The exposures that would be needed to cause internal concentrations where you're seeing activity in these cardiovascular related assays, those exposures are the same, if not lower, or if not higher than the exposures where we're actually, seeing data from, for example, the National Air Toxics database. But there were some other compounds that weren't as well, the connection to cardiovascular toxicity wasn't already well established. So things like flame retardants, certain pesticides, where there's some limited epidemiological data, but doing a workflow like this really starts to substantiate those hypotheses around, wait a second, these chemicals might be really of concern for things like cardiovascular toxicity, and they need to be studied further. And there's been a number of other applications of IVIVE that I don't really have time to go into detail, but I wanted to pop up these references in case you're interested in diving deeper. The Carlson et al. Paper looked at thyroid relevant in vitro assays and activity of pesticides in those assays, and compared the equivalent administered doses, derived from the in vitro assays. How do those compare to the POD values from in vivo animal studies that are usually, serve as the basis for risk assessments, and found that the in vitro data provides a more health protective approach to deriving those PODs. This Hines et al. Publication is actually a really nice, just kind of like how-to guide to use some of those PBPK and IVIVE models within ICE. And then this Chang et al. Publication is particularly exciting to me. It just won an award from the Birth Defects Research Prevention Society as the paper of the year for 2022. And we took a induced pluripotent stem cell assay with a metabolomics readout that predicts developmental toxicity of compounds, applied that IVIVE to it, and studied a series of valproic acid analogs, which is a sort of a prototypical human developmental toxicant. And one of the key findings here is that the dose that was derived from that stem cell-based assay was actually a better predictor of the human clinical dose where developmental toxicity occurs for valproic acid than the low effect levels from the rat developmental toxicity study, which were not protective. They were actually above the dose where we were seeing human effects in the clinic. So that was a really interesting finding. Okay, sort of a final example I want to walk through of augmented intelligence approaches that really is intended to support all of the applications I've already discussed is ways in which we're trying to automate the curation of study data from the vast number of papers that's published in the scientific literature. Historically, building reference data sets that can be used to train and test models really relies on tremendous manual efforts. And so we're trying to automate every part of that process. So use natural language processing and machine learning models to automatically identify high quality studies in the literature, programmatically extract the relevant information from not only the text, but also the tables and the figures, and then automate the annotation of that extracted information using controlled vocabularies so that we can build data sets that are not only comprehensible by humans, but also by machines, right? And we have a collaborative that's just been launched. It's being led by NIEHS and the EPA, but we have many collaborators across academia, NGOs, other Fed agencies like FDA, and it's called the Environmental Health Language Collaborative. And so you can see, so the mission is to kind of promote best practices around interoperability and harmonization of data. But we have a number of kind of use cases that have been identified here and that are being led by folks either on the federal level or some of our academic collaborators like Chirag Patel from Harvard University that I know Vasilis has collaborated extensively with. And so if you're interested in being engaged in the Environmental Health Language Collaborative, I would strongly encourage you to reach out to Stephanie at NIEHS. Okay, I probably don't really have time to go into depth into these last couple of examples of work that we're doing, but just suffice it to say that we are trying to do things like incorporate population variability into our computational models. We're doing that specifically around variability that's associated with metabolic enzyme expression across the population. So different people metabolize different types of chemicals faster or slower, depending on their CYP activity and expression levels. And so we're representing that in our computational models. We're also taking a geospatial modeling approach to kind of go all the way from sort of source to outcome, connect molecular perturbations that are identified through the high throughput screening data to known exposures like the National Air Toxics Assessment Database, and identify chemicals that have common molecular targets that people are being exposed to, and then predict the risk of that molecular perturbation at a geospatial level. So this paper actually was just selected by NIEHS as one of the intramural papers of the year. And I would highly encourage you to check it out. It's a really a nice proof of concept on how you could apply that workflow to go from molecular perturbation all the way to risk at a population and a geospatial level. So I think, one of the take-home messages that I'd like to impart here is that I feel like the future of toxicology and environmental health sciences relies extremely heavily on computational tools and augmented intelligence. So hopefully most of you have heard of FAIR. So data objects should be findable, accessible, interoperable, and reusable. And, building FAIR data objects and applying these AI-enabled approaches, I think is really the future in terms of designing iterative experiments and models that are going to give us insight into potential chemical impacts on human health and disease pathways. And there's so many exciting developments that are happening every day that if we harness them appropriately, can really support this. So large language models like GPT-4, work that's ongoing with our partners at NCATS to develop digital twins that pair in silico models with in vitro systems and in vivo human clinical information like electronic health records. So this to me is really the future of environmental health sciences and toxicology. I will come back to this slide. I'll go to my acknowledgement slide first, and then I'll probably come back to this slide because I think it's a nice one to have up during our 10 minutes of discussion that we have left. I've tried really to highlight here some challenges and some opportunities that span, scientific, operational, legislative, technical topic areas. But first, let me just go to my acknowledgement slide and just say that I'm super privileged to be leading this amazing group of individuals. The folks that are pictured all together are folks that work for Initiative, which is a CRO that provides the support contract for NICEDM. So these are a mix of administrative and scientific support staff. And then all the people who are pictured individually are part of my federal team on the NIEHS side. So I'm going to go back to this slide and then stop here. And we have about 10 minutes left, hopefully, for discussion.
We might have less. Mark, go ahead.
So thanks, Dr. Kleinstreuer, for the awesome talk. We only have time for one question because there's a class after this. I could check on the chat.
Sorry about that.
Does anyone else have a question in the class?
I have one question.
You have a question.
I'd like to ask about VCB chemicals, the chemicals that we are not aware of their structure. And one more question is about the chronic toxicity. I mean, the long-term LC20, for instance, what would the effect? And if you have worked on these two cases.
Great. Yeah. Two super easy questions, right? I'm just kidding. No, those are like really fantastic questions because those are some of the hardest things that we struggle with right now, right? Like the models that I was highlighting today are really only applicable, like the computational, the QSAR models are only applicable if you know the structure. UVCB substances are, I think there are a lot of really, really exciting platforms like iPSC derived platforms. There's like lymphoblastoid cell lines that represent population diversity, across many different donors that can be used to screen UVCBs and help to kind of rank them in terms of their potential toxicity. I think also small model organisms like zebrafish, Drosophila in particular, because many of the UVCBs are also volatile compounds. And so they're not necessarily amenable to things like in vitro testing. And so you have to come up with better exposure scenarios and like more realistic exposure scenarios. And there's a really, really cool new project that's being spearheaded by NCI, the National Cancer Institute, to develop a humanized Drosophila panel. So these flies that are being genetically manipulated to represent population diversity across, humans. And they can be used to screen volatile chemicals in UVCBs with a number of different functional and phenotypic endpoints. So I think that's where the majority of the focus is, and I think the majority of the promise is in terms of being able to address like more complex substances with ill-defined structures is in some of those platforms. And then your other question about more long-term chronic endpoints, that's where I think something like, I didn't talk about it today, but I talked about it in my lecture last month, the adverse outcome pathway framework. So developing AOPs and AOP networks that can connect molecular initiating events to key cellular events and tissue level events that basically represent our understanding of disease mechanisms and how they can be susceptible to perturbations by exogenous chemicals will help us build integrated testing strategies that represent those key biological components of multifactorial diseases like cancer, like developmental toxicity. And so we can build integrated testing batteries that cover enough of the relevant biology that we can screen chemicals for their risk for chronic diseases. But then also in terms of like looking at low dose chronic exposure over time, that's an area where I think microphysiological systems has an enormous role to play. So having tissue chip models that you can actually maintain for not only for weeks, but up to months at a time that are more physiologically complex and more relevant to human biology will allow you to do things like do chronic exposures that are more representative of what the human population is experiencing.
Okay, Nicole, unfortunately, we have to.
Thank you so much.
We have to stop because there is another class. It's our chronic problem here with all the seminars because we are in a very tight schedule. Nicole, thank you so much.